Thank you all for coming this morning.
Can you guys hear?
Yeah, I can speak louder as well if that helps. Yep. Just need to get some confidence to get going, I think. So as Tianjing said, I'm all the way from Australia, and often there's this perception that Australians are very laid-back, cruisy vibe associated with them, and I'm always trying to fight against that vibe because I think we're just as hard-working as Americans. But when I found out that I was having to give an 8:30 a.m. seminar, I realized suddenly, well, we actually really are laid back because I've never actually done a seminar or been to a seminar in Australia prior to midday. So this is quite a novel feat for me. And I heard from Tianjing just then that some seminars are also at 7 a.m. So I'm glad I didn't have to deliver that one. So as Tianjing said, I work at Monash University at Cochrane, Australia. And so for the last, I've been working in the field of systematic reviews and meta-analysis for the last decade, and I thought it would be a good opportunity to bring together some ideas of some new changes that I've seen over the years in systematic reviews and some innovations in methods that have been developed. But first of all, I just want to acknowledge some people. So my colleagues at Cochrane Australia and Monash, including Annie Sinnott, Julian Elliott, and Tari Turner, who helped me put together some of these slides that you'll see today. Also a large team co-led with David Moore from the University of Ottawa and Doug Altman from the University of Oxford. And also my funding from the National Health and Medical Research Council of Australia via an early career fellowship, which funded my visit here to Baltimore. So I'll just start off with a brief background on systematic reviews, and I'll be structuring my talk around three core issues that I see as challenges for systematic reviews, that being timeliness, transparency, and reproducibility. And so I'll explain what I mean by those terms, evaluate how systematic reviews are faring on these fronts, and then propose some solutions to some of the challenges that these issues raise. And then I'll provide a brief summary at the end, and then we can open it up for discussion. And if you have any questions throughout the seminar, please don't hesitate to interrupt me at any point. Okay. So it was back in 2007 that I started working for Cochrane, and I just came out of an undergraduate degree in psychology, and I'd never really heard of systematic reviews prior to getting my job at this centre. And it was quite easy to catch on to the notion that not only Cochrane and other organisations that routinely publish systematic reviews, including the Agency for Healthcare Research and Quality and the Institute of Medicine standards for systematic reviews, they routinely claim that systematic reviews should be up to date, that is timely, provide timely evidence for decision makers. They should clearly present what methods were used and what results were found, that is, be transparent, and they should be reported in enough detail so that others can reproduce the methods used and therefore recreate the findings. So that's what I mean by reproducible. And now, of course, systematic reviews should be other things as well, such as answer questions that are important to patients. But these are the type of things that I've had most expertise in at the moment. And I think you could devote a whole seminar on answering important questions in systematic reviews. But I'll park that issue for now and focus on these factors here. So I'll start off talking about the issue of timeliness of systematic reviews. So as many of you who are maybe working in systematic reviews or just conducting research on any particular topic, you'll be quite aware that we're facing a deluge of evidence from individual studies. So hundreds of thousands of studies are published and indexed in major bibliographic databases such as Medline every year. And because of this deluge of evidence, it's becoming harder and harder to synthesize the results of all these studies and keep up with all the evidence that's coming about. And the whole purpose of systematic reviews was originally to help clinicians keep up with this overwhelming amount of evidence. But now we're finding that it's harder even for systematic reviewers to maintain the results of systematic reviews and keep them up to date and relevant for decision making. And so there's evidence that systematic reviews do take quite a long time to produce. So an evaluation published in BMJ Open earlier this year looked at all systematic reviews registered in the Prospero Register of Systematic Reviews and followed them up for publication to see how long they took, and found that on average they took about 16 months to complete. And so, but it also ranged up to almost 44 months from the time at which a systematic review started until it was actually completed. Now, because there is this time lag between running a search and identifying studies and then actually publishing the results of all those studies, we find that systematic reviews quickly go out of date. So by the time they're published, there's often this time lag between what evidence is actually included in the review and some subsequent evidence that may have been published. And so in a study by Shijana published in Annals of Internal Medicine a few years ago, they found that within 2 years post-publication of a systematic review, about a quarter of the reviews had an incorrect conclusion because they weren't able to, well, they didn't incorporate all the recent evidence that had come out in those 2 years, which may have led to a clinically meaningful difference in the results. Now, one way to deal with this, this currency challenge for systematic reviews that has gained quite a lot of traction, especially in the last 6 to 12 months, is this concept of having living systematic reviews. And so my colleague back at Monash University, Julian Elliott, published this paper back in 2014 where he coined this term of living systematic reviews, which I'll just provide you a brief introduction to. As a potential solution to the problem of timeliness of systematic reviews. And so what do we mean by a living systematic review? So essentially a living systematic review, or LSR, is a systematic review that's continually updated, incorporating new evidence as it becomes available. So the key elements of a living systematic review are that, one, it is a systematic review and retains the core methods that a review would use, but also it is different to other standard reviews in that it is continually updated, whereby authors need to perform monthly searches of the evidence rather than the standard approach whereby a review is not updated at all or may only be updated every few years. And as well as searching continually for evidence, it also incorporates the findings of those new studies and synthesizes the results of those studies. Now, in theory, this living systematic review approach could apply to any systematic review on any type of question, whether it be looking at the effects of healthcare interventions or the diagnostic test accuracy of a particular test. But it would first start off with the authors doing a standard systematic review, which is what we would call the baseline review. But then, unlike the normal practice where you publish your review and then move on to different projects and completely ignore this work that you've been doing, you would then have continual evidence surveillance, so continual monitoring of biomedical or bibliographic databases to see what new studies are published, ideally continual incorporation of that evidence and dissemination of the findings of those continually updated systematic reviews. So rather than just publishing one article, you're constantly letting the public know what the latest findings are regarding the effects of, say, an intervention or a particular test or the prevalence of a particular condition. So some of you who might be quite experienced in doing systematic reviews thinking, who the hell would even consider doing a living systematic review, and why would anyone be motivated to do this, given there's so much workload involved in doing just a standard review? And so we think that there's a couple of reasons why you might consider doing this. If you're looking at a question that is a really high priority for policy and practice, then it may be you may consider it necessary to continually update the findings of your systematic review so that it maintains relevance. Also, if you're looking at an area which is quite a fast-moving area of literature where there's a lot of important uncertainty in the existing evidence, and so every new study that comes out comes with a conflicting finding about whether a particular treatment works or not. Also, if there's emerging evidence, for example, in clinical trial registries such as clinicaltrials.gov, that suggests that it's likely that once that data from that trial is included, it's going to have a big impact on what we know. And so as an example of one sort of high priority question that meets this criteria is several reviews that have been turned into living systematic reviews around the Zika virus, led by Nicola Lowe from the University of Bern. So this is an example of a review that was registered in the Prospero database of systematic reviews, which is looking at the sexual transmission of the Zika virus and other flaviviruses. And so she's been doing several other reviews focused on the Zika virus, where there's obviously a need to try and find accurate evidence in a very timely manner. But also another thing you really would have to consider is that you and your network of contributors actually have the capacity and resources to sustain an ongoing commitment to this living systematic review. So you need to have the manpower and team of people involved who actually want to do all the work to keep this review up to date. Now, the Cochrane has a living systematic reviews network who have been doing quite a lot of work in the last year or so in developing guidance for living systematic reviews. And just the last couple of months, they've published on this website listed at the bottom here some interim guidance for authors considering doing a living systematic review, because they've just recently started an initiative to pilot the work of actually doing these reviews. And they have a template for authors to follow, providing them with lots of guidance on how they would go about doing these reviews. And I'll just outline some of the core methods of doing these reviews that are currently proposed by this network. So in terms of searching for the evidence, there's an expectation that you would be quite explicit about how frequently you're going to be searching for any new evidence on the particular question that you're focusing on. So ideally you would be searching electronic databases such as Medline or Embase and your trial registers to find new studies on a monthly basis by using potentially auto alerts for your searches. And you may also be searching other sources, such as websites of government reports or conference proceedings, but perhaps searching these on a case-by-case basis, because it may not always be completely relevant to search all of these additional sources on a monthly basis if very little evidence is actually added to those databases on a routine basis. And you need to rerun each of the searches in full, but it would also be advised that people would be evaluating the search strategies they use over time to make sure that the terms that they're using in their line-by-line search strategies actually are relevant and more likely to keep picking up the most relevant research. Now, once they find these new studies, authors could just go about the standard approach of actually assessing each article for eligibility, so screening the records they obtain, and then also collecting the data from those particular articles. But they may also consider using some novel tools that are starting to gain traction at the moment to help speed up the process of doing these reviews, including machine learning and using participation of citizen science crowds. So some examples of some machine learning or semi-automation tools that authors may consider using. One of them is a project launched by Cochrane known as the Evidence Pipeline, which is using text mining to classify medical articles, first of all, into the type of study design that is used in the article, whether it be an RCT or a diagnostic test accuracy study, then picking the topic area or classifying the clinical topic area, such as cancer, back pain, or heart disease, and then also recording the main PICO characteristics of the trial, such as the participants, intervention, comparators, and outcomes, to see whether a particular article is most likely to be of relevance to a particular review question. And other tools such as Robot Reviewer are using machine learning to help authors extract information from published articles and to assess the internal validity or risk of bias of the included studies so that they don't have to manually do it themselves. And so these tools are starting to be piloted as a way to help speed up the methods of doing a systematic review. In addition to the more technological methods, there are other methods people are experimenting with, and one of them is participation of a crowd of contributors. So Cochrane has a voluntary crowd known as the Cochrane crowd, including about 6,500 contributors who, on a voluntary basis, are willing to look through published articles to classify them as being either RCTs and what sort of clinical area they belong to. And so these include a lot of medical students and people of any age really who are interested in helping authors be able to conduct systematic reviews in a timely manner by doing this voluntary work. And there's also a website called Cochrane Task Exchange, which has been described as a dating site for systematic reviewers, which essentially allows you to get in touch and advertise particular projects to help push your systematic review along. So you can just say, I'm working on a review on diabetes, I need some help screening, does anyone want to help me? And they can get in touch with you and say, yes, I'd like to help. And in acknowledgement, they may be come on as authors of your particular paper. But we're finding there's a really generous community out there who are willing to help people get syntheses of research done. Outside of Cochrane, there are other initiatives that are requesting participation of crowds of researchers. So in the Lancet Neurology last year, there was a call for researchers to join the Meta Microbleeds Consortium to help push forward systematic reviews on cerebral microbleeds. And a team in France have also established a website, live-network-meta-analysis.com, which is asking for people to contribute to a cumulative network meta-analysis on second-line treatments for advanced lung cancer. So anyone who's interested and will undergo some training was able to help contribute to getting the results of systematic reviews done and up to date in a timely manner. Now, once you've actually found the evidence in your living systematic review, there then needs to be the decision about when you would incorporate the findings of those new studies. And so the ideal default position would be that anytime you find a new study that's relevant to your review, you would incorporate those findings, you'd incorporate the data and the information in your review. But of course, there may be instances where you do find a study that's very small or potentially very likely to be at high risk of bias, where it's unlikely that including it in the review is actually going to change the findings in a meaningful way. So authors would be advised to come up with some decision rules or strategies to help them decide when they would actually incorporate new evidence as it's identified in these searches. Now, one main core challenge of doing a living systematic review is the fact that you're frequently updating meta-analyses. And so when you consider it, if, say, you were finding a new study every single month to add into a meta-analysis, you are having to recalculate the meta-analytic effect each month. Thereby, performing your meta-analysis multiple times means that you're inflating a type 1 error and increasing your chances of false positive results. You're increasing the chance of saying that there is an effect when there actually isn't. Now, this applies to all systematic reviews, but is obviously more of a problem for living systematic reviews if you are in that situation where you're looking at a very fast-moving field where lots of evidence is continually added to the meta-analysis. And so several methods have been proposed to adjust for this frequently updated meta-analyses, but at the moment there's not really clear consensus, one, on if we should be adjusting, and if so, how. So some of these methods include trial sequential analysis or sequential meta-analysis, but some of these studies are essentially have rules that are so strict that they add corrections to like either the confidence bounds for a meta-analysis that no matter how many times you update the meta-analysis, you'll never detect a statistically significant result because it's so conservative. And so there's a project being led by Mark Simmons at the University of York in the UK, which is running some simulations to evaluate each of these adjustment methods to see which one fares the best. And so we'll ultimately be getting some guidance on that, hopefully quite soon. Now, the whole changing landscape of doing systematic reviews in a living format has meant that there's needed to be adaptations to how we actually publish and disseminate research. Because originally, the standard approach is obviously you would do your systematic review, submit it to a journal, wait for several months for it to be peer-reviewed, then have it maybe accepted and have several more months waiting for copy editors and then having the article finally published. Now, obviously, all those time delays are not going to be helpful if your aim is to actually get the evidence out there quickly. And so some journals are experimenting with new strategies of disseminating research. So the Journal of Neurotrauma, who published this living review on adherence to guidelines in adult patients with traumatic brain injury, has now committed to essentially having an continually updated appendix or supplementary file for this review. So whenever authors contact or try to access the initial file, they would immediately be sent to a link that takes them to the supplementary file that tells them what the results of the monthly or maybe a bit more than monthly update of this review find, showing what new evidence was included and how the results have actually changed. Other people are experimenting with actually just disseminating the findings of their living systematic review online. So this review published in PLOS ONE last year looking at screening coverage needed to reduce mortality from prostate cancer. The authors of this review have created a website on GitHub where they're actually going to be disseminating the results of this, this updated review on this website whenever the new evidence becomes available. So several different strategies are being used to sort of test how we disseminate this research. At the moment, my colleagues led by Annie Sinnott in Monash University are doing a more formal piloting of living systematic reviews in the format of Cochrane reviews, where there's 4 author teams at the moment who are piloting their Cochrane review using some living systematic review methods devised by the network, the LSR network, supported by the team at Monash. They are using machine learning and other things like the Cochrane crowd to see how feasible and appropriate these methods actually are at getting living systematic reviews done within the context of Cochrane. And I think the first reviews would have just been uploaded in the last few weeks on the Cochrane library, and we'll be able to get some good data on how feasible and valuable these reviews are over the coming months. And this whole enthusiasm for living systematic reviews is now expanded beyond systematic reviews to clinical practice guidelines. So the American Society for Hematology and the Brain Trauma Foundation are both piloting methods for actually keeping the results of clinical practice guidelines up to date as well on a regular basis. So rather than using a more standard approach of updating the guidelines every few years, they're keen to try and keep the evidence up to date on a much more frequent basis. So there's been a growing enthusiasm for living systematic reviews, especially in the last 6 to 12 months, and they're considered to be highly desirable to evidence users who don't really find it that helpful to have a review that is several years out of date being able to influence their practice. But there's obviously still needs to be work to establish the feasibility and implication for authors, publishers, and users of these reviews. We also obviously need to resolve the issue of frequently updating these meta-analyses and use of statistical adjustment methods. And also we need to give some thought about when a review will no longer need to be kept living, because it's unlikely that you'll need to keep on updating the searches and trying to find evidence forever. Because sometimes the evidence just slows down the pace in which new evidence actually becomes disseminated. Some people too have also been a bit alarmed by the term living systematic review because they start thinking, well, what do we call our standard systematic reviews now? And I know some of my colleagues back home consider their systematic reviews to be roadkill, but I don't think that's necessarily a good approach to thinking about your review if it's not living. But there's a lot of activity at the moment as groups experiment with these methods, and it'll be exciting to see what they find in the next year or so. So that was just the first issue I wanted to talk about in systematic reviews. And now the next one I want to move on to is talking about issues of transparency of systematic reviews. Now, regardless of whether you are doing a systematic review that is living or not, obviously users need to know what you actually did and what you actually found in your review. And so there was a great paper or a great series on research waste in The Lancet a few years ago. And this article by Paul Glazou and colleagues looking at, provides many pointers as to why incomplete or unusable reports of biomedical research is just quite harmful for users of evidence because they're not able to reliably understand the results of the research, to judge whether the research is conducted using valid methods or not. And so there's many good reasons why we want to be clear about what we've actually done when doing a systematic review. And so one way to help increase the transparency of systematic reviews is to use reporting guidelines. And so in June 2009, some of you or many of you may be aware of the PRISMA statement, so the preferred reporting items for systematic reviews and meta-analyses reporting guideline that was disseminated, which has, in terms of citations, has had a major impact on the systematic review community, being cited more than 20,000 times since 2009, endorsed by 180 journals and editorial organizations, and it's actually spawned a whole series of extensions of reporting guidelines to provide guidance on different types or aspects of systematic reviews. So for example, there's an extension on network meta-analyses or on reviews focusing on individual patient data meta-analyses. And there's 8 even in development looking at different aspects of reviews. Now, the lead author of this, David Mower, and myself were interested in whether PRISMA is actually having its intended effect. And so is it actually leading authors to report their systematic reviews more transparently? And so in some work that's under review, we did a systematic search for studies evaluating how well systematic reviews adhere to the PRISMA guidelines. And so we searched Medline up to July of this year for any studies evaluating adherence to each of the items in PRISMA in systematic reviews that were published in 2010 or later. And so we picked 2010, given this was a year after these guidelines were disseminated. And so we wanted to see how these recent reviews are faring in terms of how well they report their methods and results. And then we pulled the number of reviews that adhered to each item across all the studies we found. And so after initially identifying about 5,000 records, we found 27 studies that have looked at systematic reviews in different clinical areas, a total of 2,300 reviews, and evaluated them against the criteria in the PRISMA statement to see how well they were reported. And so this figure here is obviously a little bit hard to read the text, but it reports the proportion of reviews meeting each of the criteria and the 27 criteria in the PRISMA statement. And so first thing you might be able to notice is that none of the items have 100% adherence, but there is quite a bit of variability across the items at how well authors report the methods and results that they've actually used in their review. And so when you order them from most to least reported, we see that there's about 15 items, so just over half of the items are adhered to by less than 80% of published reviews. Then we also have 8 items that are adhered to by less than 2/3 of the reviews. So issues with reporting risk of bias, how they actually search for studies, any additional analyses such as subgroup analyses, funding, and having actually a protocol or registration of their methods have not the best adherence in in this particular sample of reviews that have been evaluated against this criteria. And so based on this and other research, we've considered that it's now a good time to consider updating the PRISMA statement to help increase the transparency of future systematic reviews. And so there's a couple of reasons why we think an update of PRISMA is needed. So there are, of course, 27 items in the checklist, but some of them include really a large number of components within themselves. So if you even look at just item number 7, which asks authors to describe all information sources, databases with dates of coverage, contact of study authors in the search, and the date last searched, when you actually break that down, that's actually getting people or prompting people to describe 4 different things. 1, which databases were searched, such as Medline and Embase. 2, whether they actually contacted trialists to identify additional trials. 3, what the dates of coverage of these databases were. So were they looking at Medline, studies in Medline from inception to 2010 or from 2000 to 2010? And also the date in which the last search was conducted. So potentially rephrasing or breaking down these multi-component items might be a better way to increase adherence to get all this information documented in a review. And also, given it was published in 2009, there's been a lot of changes, obviously, in methods for systematic reviews that have happened in the last 8 years that potentially need discussing with a wider group of stakeholders to see whether they could and are worth adding into an update of this guideline. So at the moment, there isn't really much guidance on what authors should report when they come across multiplicity of outcome data in studies and how they dealt with that multiplicity. There's very little guidance and information on how you should report the characteristics of the interventions of studies included in your reviews, such as their dosage, duration, how many people actually delivered the intervention. And there's been really interesting work from Tammy Hoffman published in the BMJ, I think a month ago, which lays out a quite nice proposal for how authors could actually report their interventions of each included study that could be incorporated in an update of PRISMA. There's very little to say about the use of prediction intervals when interpreting random effects meta-analyses, and little to say about how to enhance the reproducibility of meta-analytic results and share the data that has been collected in reviews. And also little guidance on reporting methods and results of updates of systematic reviews and living systematic reviews. So I'm not saying that all of these may be included in an update, but they're worth considering. But as well as talking about what items to include in a new reporting guideline for systematic reviews, it's also worth developing new tools to increase adherence to these guidelines. Now, if any of you have actually used these guidelines, such as PRISMA or Consort, you'll probably recognise that these checklists are often used and thought of after the fact, because most times you'll spend painstaking hour after hour with your team of authors to write your article, and then you go and submit it to a journal and suddenly realise that the journal says, can you send us your PRISMA checklist or your Consort checklist? At that point, you're less willing to make the modifications to the article, and you just quickly rush through and fill out the checklist, and God knows whoever actually reads the actual checklist that's submitted. I don't know if the peer reviewers actually pay much attention. So tools to help journals and authors actually better adhere to reporting may be a smarter approach. And so last year, or a couple of years ago, a team in France developed some online writing software that they called Cobweb, which could be used at the start of writing a randomised controlled trial report, and provided guidance along the way on how to report their trial report in adherence to the Consort statement. Developing something like that for PRISMA may be a smart way forward. And a number of startups have been developed over the last couple of years. So Stat Reviewer is one of them, and Penelope Research, who are working with journals to help them essentially develop software so that once manuscripts are submitted to that journal, they scan through them to check how well they actually adhere to the reporting guidelines, or whether there's any statistical errors within these articles. And so that's a good secondary check that takes the effort out of human editors and peer reviewers having to do all of that checking. So working on that in the context of an update of the PRISMA reporting guidelines, we think is worth trying out. Now, the final thing related to transparency that I want to talk about and how that's going in the field of systematic reviews is this issue of reproducibility of reviews. And so, unless you've been living under a rock, over the last few years you would realize that we are currently in the throes of a reproducibility crisis, where we have lots of studies and commentaries and debate and editorial articles telling us that the findings of research are just not reproducible. When someone tries to recreate the findings of a particular study, whether it be in psychological science or cancer biology, we're finding that the findings of these replicated studies don't necessarily match up with the original article or the original study that was published. And so my colleague collaborator David Moa, had the great idea of thinking, well, maybe we should actually, rather than continually criticizing individual studies such as randomized trials or individual experiments, maybe we should hold systematic reviewers to the same account and see how well they are at producing reviews that are reproducible. And so, because we think there's a number of positive aspects of actually using reproducible research practices, because they essentially allow others to recreate the findings of your studies given the data that are available. So they can be used in the context of systematic reviews to allow for reanalysis. So if you see a meta-analysis published and are interested in only a subgroup of those studies, if you have the shared data available, you potentially could do your own subgroup analysis on a set of patients or set of studies conducted in the setting you're interested in. They also allow for checking of any errors in the published review, and also they would allow others to cumulatively add new data once it's published. So in other words, create living systematic reviews based on the shared data that is available. But as I said, it's not really clear how often systematic reviews, particularly of therapeutic interventions, actually follow practices that would result in reproducibility. So in some other work done with Doug Altman, University of Oxford, and Larissa Shams here from Ottawa. So last year, a team of us published a paper in PLOS Medicine looking at the prevalence and the reporting quality of systematic reviews of biomedical research. And so what we specifically looked for were all systematic reviews indexed in Medline in a single month, being February 2014. And in that initial study, we found about 680 meta-analyses or systematic reviews in that particular month. And so for this sub-study, we focused on just the subset of 110 reviews that were looked at the effects of therapeutic interventions, being either treatment or prevention questions. That included at least one meta-analysis, and we didn't have any restriction by journal or clinical specialty. So Cochrane and non-Cochrane reviews and any clinical condition that was found in those reviews we included in our analysis. And we had one author evaluate 80% of these systematic reviews, and 20% were evaluated in duplicate to check for any errors that may have been made in evaluating these reviews. And some examples of things that we looked at when we were evaluating the reproducible research practices were we wanted to see whether the authors reported enough detail so that others could actually recreate the meta-analyses. And I'll show you what I mean by that on the next slide. We also wanted to know if authors not only state if they actually obtain unpublished data from trialists, but which specific data they actually obtained. So too many systematic reviews at the moment will just include a statement such as, if data were partially reported or unclear, we contacted the authors. That's all they'll say. They won't tell you if they're actually successful in obtaining any data, and if you were to go and look at a meta-analysis on a forest plot, you would have no idea which, if any, of that data comes from the authors rather than the published paper. And we also wanted to know if authors are making the data sets and the data that they've collected and the statistical analysis code available for others to use in reanalysis efforts. Now, in terms of the ability to recreate a meta-analysis, seeing if they reported the required information, things that we were actually looking for was whether for each meta-analysis they reported either the study, well, they reported one, the list of studies that were included, the treatment effect and, say, a confidence interval for each study, the final meta-analysis result and its precision, and the model used. Now, looking at those 4 criterion under that first, and that green box there, some of you may be thinking, well, clearly all of this information is available on a forest plot, so all meta-analyses should be reproducible. But that's not always the case. So some authors sometimes only report for you what the meta-analytic effect is, or provide a narrative description saying, for example, our meta-analysis of pain was not significant, whereby you don't know which studies were included and which data from each study was actually included in the review. So across the 110 systematic reviews in our study, where there was a total of just over 2,000 meta-analyses that we evaluated to see if, having a look at them, you could actually recreate the data. And we found that an okay majority, so 73% of these meta-analyses could be recreated from the data reported. But still, about 588 meta-analyses, it would be impossible to attempt to recreate the findings because we, for example, had no idea which studies were included and which data had actually been included from each study. Now, when you actually look across the review at the review level, so across all 110 systematic reviews, we found that the reviews reported the data needed to recreate all meta-analyses in only 65% of the cases, but they were of course more likely to report the data needed to create the primary meta-analysis of the review. It's just that they weren't always reporting it for all meta-analyses that they conducted. If they'd done any manipulation of the data and stated that they'd used some manipulation of the data, such as combining 2 groups in a 3-arm trial or converting or imputing any missing standard deviations, it was really only clear which particular data were manipulated in about 40% of reviews. Those who said they retrieved data from trials, in only about 40%, they specified which data actually had been obtained. And only about 30% made their data sets and the raw data they'd collected and the analysis code available. And essentially that was slightly being generous there when we made this assessment, because this pretty much applied just to the Cochrane reviews, which are published with a review manager file that does include editable data and code to actually rerun an analysis. It's not the most ideal scenario though, because it's not using, for example, an Excel file with maybe starter or SAS code that would be much more easy to transfer across different platforms to allow reproducibility. And now looking at whether there was a difference between our Cochrane and non-Cochrane reviews, we found that there was quite a big difference in how well the information was reported that would facilitate these reproducibility. So only about 50% compared to about 95% of the systematic reviews reported all the data needed to recreate meta-analyses. Very few reported data on unpublished, on which unpublished data was accessed, and providing data sets and statistical analysis code was obviously a lot less common in our non-Cochrane reviews. But we were also keen to see if those systematic reviews that claimed that they actually used the PRISMA reporting guidelines were more likely to use reproducible research practices. And we actually found there wasn't that much difference at all. So those that said that they followed PRISMA were actually less likely to report the data needed to recreate all meta-analyses, which is somewhat counterintuitive. But if you actually take a close look at PRISMA, you'll see that it's not completely explicit in terms of how much data you should be reporting in your review and making available. So it's maybe not that surprising that there wasn't much of a difference between those who said they followed these guidelines and those that didn't. Now, in the only other study that I'm aware of that has looked at reproducibility of meta-analyses, this was done by Daniel Lakens this year, looking at meta-analyses in psychology. So they looked at 54 meta-analyses in 3 psychological journals in 2013 to 2014, where they found that only 67% reported the data needed to recreate meta-analyses, which is quite similar to our estimate of about 73%. And they actually went one step further than us by actually randomly selecting 20 of these meta-analyses and following all the steps stipulated in those published reviews to see if they themselves could reproduce the findings. And they first found that 5 could not be reproduced at all because of lack of raw data. That is, all data in the meta-analysis had come from the authors and was not in the public domain. There were no details about the effect sizes for each study or lack of information in how effects were coded. And then in the other 15 meta-analyses, there was differences between the reported and reproduced effect sizes and sample sizes for the included studies. So these were quite common. So an implication for enhancing the reproducibility of results of systematic reviews could be to make greater use of software to share the meta-analysis results and the underlying data. So software such as the Systematic Review Data Repository, which I know many of you in Johns Hopkins are familiar with and are making great use of to store the data that has been collected, as well as other platforms such as the Open Science Framework or Figshare to be used to actually share all these results that you have actually conducted in your systematic review, which may not necessarily be able to fit within the published article, but could easily be linked from the article to these external websites. And some people are also working on R and Markdown to help better link the information that has been collected from studies so it's easily seen where that comes from when you're actually looking at a published article for a systematic review. So just a quick summary of all those things. So obviously what we want with systematic reviews is them to provide credible evidence for healthcare decision making. But we do have some emerging evidence that there are many systematic reviews that are either outdated at the time of publication, they fail to adhere to existing reporting guidelines for reviews, and they fail to report their methods and results in a way that allows users to reproduce them. And so I've just outlined today some potential solutions that are being trialled out at the moment to help overcome these challenges. So either using living systematic review methods to aid timeliness of reviews, revising PRISMA and developing technology to facilitate adherence to these guidelines to enhance transparency, and encouraging authors to make greater use of data sharing platforms to aid the reproducibility of systematic reviews and meta-analyses. So I think I will stop there and welcome all your questions. So thank you.
We have about 5 minutes for questions.
Yeah, you haven't mentioned anything, I don't think, about individual bias in some of these areas. For the living SR, a new trial comes along and it will change the conclusion. Now people can either look for all kinds of reasons not to include it, or say, well, we have to wait for still another trial to be more persuasive. And maybe that is the best thing to do. I think also with reproducibility, once people know the result, okay, I know the result of what the other people did, I want to see if I can reproduce it, they can find ways to include other trials or eliminate trials to get the result that they want.
Yeah, well, yeah, that's a good point. These things obviously do need to be considered in terms of helping ways to overcome that bias as well. And hopefully, I would say if you were going to be doing these reviews, you would obviously be pre-specifying before you'd actually seen the data what sort of methods you'd be used to decide when to include that data. But yeah, those things obviously do need to be considered.
So thank you for outlining some of the techniques that would allow for more timeliness and reproducibility and so forth with systematic reviews. My question is, does the current incentive structure for researchers with the need for numbers of publications and impact of those publications encourage using these techniques?
I think the current incentive structure does not encourage these techniques, but I think we obviously need to move towards a situation where it does, because I obviously understand, particularly more in the context of the living systematic reviews, where you may only be having the one published article and then maybe disseminating your findings on websites. That's clearly not going to get you the same, same credit in terms of your CV. And so there are ongoing discussions with different groups on how to go about that. And I think even within Cochrane, they're still discussing, well, do we give a new citation for every single update of this particular review or not? And I mean, maybe if we move towards a world which actually does provide incentives for sharing of data rather than just number of publications, maybe that is one way to tackle the problem. But I do recognise it's a bit of an uphill battle to get people to use these methods at the moment.
So you mentioned using living systematic reviews for guideline development, and guideline developers typically start off with some big stakeholder constituent group that they want to involve in the process, and they're going to review the data as it comes in and talk about what the implications are. Cochrane and other journals, Cochrane in particular, has a very onerous editorial process where the editors and methodologists have a great deal to say about what it is you're proposing to do and then how you're analyzing and interpreting your results. So how do we reconcile the desire to do these things quickly on an ongoing basis with all of these other requirements that patients and stakeholders be involved, that editors check everything out?
Yeah, I mean, in terms of the guidelines, I'm not too sure exactly what sort of systems they're going to be putting in place for that, because obviously it does need wider consultation than just a set of peer reviewers and editors. But I know, at least within the context of the living systematic reviews themselves, they have the Living Systematic Review Network and Cochrane are trialling out different methods of having pools of peer reviewers who are actually committed to being involved with each update of the actual living systematic review, and so not having to always invite and try and find someone at each time, and not always having the same same number of people each time an update does occur, and so using a bit more expedited peer reviewing of that data, at least when you are looking at those monthly updates. But I'm not too sure exactly what they're proposing with the living guidelines at this point. So.
Yeah. So it's great that these methods are getting, for example, with Timeliness, getting the evidence out there faster. But I'm curious how you're measuring if it's doing any impact. For example, with the guideline development, right? I'm thinking of Eyes and Vision and the AAO. Those preferred practice pattern, those clinical practice guidelines come out every 5 years or so. And it's quite exciting. We look at the recommendations and what kind of evidence they cite. It's quite exciting when they cite a systematic review. But how do we integrate that living systematic review system into such the guideline development process?
Again, I'm not too sure about going beyond the living systematic reviews, unfortunately. That's where I've spent most of the work, and putting, I think it is engaging more of the systematic reviewers, so they're actually maybe not always, they're probably working much closer with the guideline development team to actually just do the continual analysis and inclusion of these new trials. But the actual processes, I'm not too sure about that. But if you have any ideas, that you can definitely pass on to the network, who are really, the only really 2 organisations are trialling this out at the moment to see how feasible it is to do this and what, as you say, what impacts it's actually having on the guidelines. And even on the reviews themselves, like, searching on a monthly basis may not really be changing the conclusions at all. So there's obviously still work in trying to test that out.
Carl?
Thank you very much. It has been an excellent presentation, particularly with the proposal of the living systematic reviews. I would be interested if you would be expanding about the use of artificial intelligence, particularly for the timeliness. And how do you see the role of co-evidence now?
Role of what, sorry?
Co-evidence.
Of what?
Co-evidence.
Covidence?
Yeah, is the use software?
Oh, Covidence software, yes, sorry. Yep. Exactly. It's actually produced in the people who sit right next to me, so in Monash, sorry. I thought you were saying computers, sorry. Covidence. So Covidence software is another platform, one of many platforms that are available to help authors conduct their reviews, at least share the tasks of their reviews. At the moment, it's not actually using machine learning approaches. It's more being used as a resource to collect data from studies studies to screen articles for inclusion. And so all the text mining work and machine learning algorithms are being developed in external software, but definitely Confidence is recommended within, at least within Cochrane, as a software that can make it easier to collaborate on these reviews. But in terms of the actual machine learning processes, I'm not an expert in that area, but as I said, they are being trialling out, and there's I could refer some papers maybe after this seminar that could give a bit more information. And I know that the Journal of Clinical Epidemiology is coming out with a series, I think probably in time for next week's Cochrane conference, which actually has a whole series on living systematic reviews, one of which is going to be on these machine learning methods that are being used in that. So a lot of the things I've mentioned will be explained in a lot more detail in Journal of Clinical Epidemiology, I think next week. So.
All right. Thank you very much, Matt. I know many students have commitment at 9:30, so we're going to conclude here.